So I'm just going to do a very short introduction to this, really as a sort of thank you to the people who've taken part in our sort of biomarker study, and just tell you how that's sort of going and what we're doing with that. Then, as I say, we're going to hear from Aru and Debbie after that. So one of the studies we're doing, and I can look at quite a lot of you and know that that you've been approached to take part, and uniformly said yes in this, is a study called the BioLAM study, which was a name dreamt up by our research nurse, but it's sort of it'll do, won't it? So the study is a biomarker study, and biomarkers are either a test, it might be a blood test or some sort of measurement or some sort of clinical factor that can either predict whether a disease is present or not, how active the disease is, or perhaps whether the disease will respond to treatment. And really the aims of the BioLAM study are twofold. To try and identify factors to reduce diagnostic uncertainty for LAM, so improving other methods of biopsy. But really the main aim is to try and identify early stage in the disease who's at risk of developing progressive lung disease and who's likely to run a more indolent course and stay well for a prolonged period of time. And as we've heard earlier, this will be useful for targeting treatment. And whenever you're applying for funding, you have to sort of put in all the sort of trendy terms that are around at the moment. And the ones at this particular time were stratified medicine and personalized medicine. So these terms, what they really mean is taking an individual's illness, not the sort of average thing that's written in the textbook, but whether there's a progressive form or a more stable form, and then tailoring their treatment and their medical care exactly to this situation rather than just the average. And that obviously improves the outcome for individuals, is better, and is also cheaper. So it's sort of an approach to stratified or personalized medicine in LAM. As we've sort of said before, the course of LAM is really variable. Now, all of these lines are the FEV1 of people that we've seen at the LAM Centre over actually some of the many years. And you can see there's people with normal lung function, there's people with low lung function, and there's millions of people in between. And if we just pick out 3 patients here, there's this person with an average rate of decline for a woman with LAM. Then there's this person here whose lung function's normal and stays normal for, well over 5 years. And then we have people with a more progressive course. And if we could predict which of these patients, which course people were going to follow early on, then we could sort of really improve their medical care significantly. We could give people more accurate information about what their prognosis was likely to be. If we knew somebody was going to have progressive or rapidly progressive disease, we could treat them much earlier and avoid them losing lung function before we make a decision to treat them. And when we're doing research, we can enrich the clinical trials with the right sort of patient. So if we have a treatment for progressive disease, we could only enroll patients with that. We wouldn't need so many people, we'd get the answer quicker. So the BioLAM study takes advantage of the people that come to the LAM Centre. Well, takes advantage is probably the wrong word, but you sort of know what I mean. Benefits from the patient cohort at the LAM Centre. So we'd already had approvals to, with ethical permission, use clinical data and get blood samples and DNA samples from people coming to the centre. And we wrote a study which aimed to recruit 150 women with LAM, and then we'd compare their imaging values, their lung function, which we measured over time, so it's useful. Multiple data on individuals, and we'd know what treatments they're on, and we could use this to develop biomarkers. And we applied to the NIHR using all these trendy words about personalised medicine, and we got a grant to deep phenotype, which is another trendy way of saying to understand the individual's illness in detail, to deep phenotype the whole UK cohort through 8 centres. patients coming to Nottingham. We will be seeing patients also in Cambridge and London in the future. And there are these other NIH centres who are actually just sending patients to us. So we've been funded by the NIHR to do this study, and it's a pretty straightforward study. We see patients, they already have had lung function, they've already had scans and other clinical data, the medical history is already in their notes. They may or may not have had a tissue biopsy previously. So we take consent from them to enroll them in this study, and we take a blood sample. And then we don't do anything very much, really. We just look after them in the normal way for a year. And what we see, of course, is that some patients have disease progresses over that time, and for other patients, the disease remains stable. And so we measure their lung function and their outcome at the end of the year. What we also have is what's happened before, their historical change in lung function, and for some patients, we have biopsy tissue. And so we use all of these data, the lung function, the clinical features at the start, factors from their biopsy and what's happened previously, and as Frank said, the best predictor of what's going to happen in the future is what's going to happen in the past, to try and predict what's going to happen over the next 12 months. And we're hoping that we'll be able to continue this study if we get further funding at the end of it. But so we're hoping that by identifying differences at baseline between the people who've had progressive and stable disease, we can design a test that will predict the outcome early on. And so once we've done this, we can go to another population and we can see if our test works and is robust and is reproducible. So that's the idea of the study. It's fairly straightforward. We're measuring a few things. You've heard about VEGF-D already, which is this biomarker that Frank has shown predicts the course of LAM in his population. Does it do the same in all comers with ours outside of a clinical trial? That's important. This molecule collagen 4 is an extracellular matrix protein that's turned over when the lungs are damaged, we think. It's been shown to be related to activity in LAM. So we're validating these. We're looking at lots of things in tissue, particularly things that have come out of research at the NIH, like the LAM histology score, but also stuff from Debbie and Aru's work, specific markers that we've identified that we think may be helpful. And we're doing various omics approaches. So omics is a modern research technique where instead of thinking, well, I think maybe this gene's abnormal in LAM, or this protein might do this because of X and Y. You just don't have a hypothesis at the start. You just look at all of them and see which ones are different. The hypothesis-based approach is like looking at VEGF-D or collagen 4. Occasionally you're right, but there are 30,000 genes in the human genome, and to get the right one is inspired. And sometimes you have inspiration, and often you don't. So you do an omics approach, and most of these are new technologies. And if you look at the labs websites that do omics, they say it's all very exciting. We'll have robotic sample handling, automatic data analysis. So we're doing various omics approaches on this. And when I heard about the robotic sample analysis, I was very excited. I was thinking, oh, this is going to be brilliant. Look at this. Just data coming out everywhere and sorts of stuff. So, I went to the proteomic facility, which is Nottingham Trent, which is at the other university, where they're proteomics experts. I was a bit disappointed when I got there, to be honest. What a rubbish robot. Anyway, but we're doing various omics approaches at various levels, which we hope will shed light in an unbiased way on factors that are associated with the progression of LAM. So that's the study, really. My main thing I wanted to do today was to say that I've never done a study before where I've asked 133 consecutive patients to take part, and 133 consecutive patients have said yes. In fact, now I'm just dreading asking people, but in case I jinx the whole thing. So thank you so much for your support and participation in this study. It's really great that everyone has said yes. Anyone who hasn't had the opportunity to say yes, of course, we're always happy to see you at the LAM Centre to do that. And the study's funded by the NIHR. The proteomics study is funded by the LAM Foundation, kind enough to give us a grant. Of course, we're continually funded by LAM Action in various capacities. Just say thank you to Janet and Sharon at the LAM Centre. The person who should have been doing this talk is this lady who's Suzanne Miller, who's funded by the NIHR to work on this project. she's at a hen night in Marbella. I know where I'd rather be out of those two places now, and it's right here despite the weather. So I'm going to hand over to Aru now.
Hello. So I'm sure everyone knows me by now, but for those of you who don't, I'm Arundhati Dongre, and I have been doing my PhD with Simon Johnson for the past 3 years. So I just would like to tell you about my PhD that has been very kindly funded by LAM Action. So my lifelong ambition, basically, has been to be a scientist, and for everyone who asked me, I said to them that I would like to be a scientist when I grow up. And so after I finished my Bachelor of Pharmacy degree in India, I decided that scientific research was what I would like to do instead of being a pharmacist. So then I flew from India to the United Kingdom in 2007 to pursue my master's and then to continue on with a PhD. So I initially came to work for Simon Johnson as a research technician, actually working in asthma. And it is then that I got introduced to LAM. And the more I read up about it, the more interesting the condition felt to me. And I thought I would like to know more about this through research. Initially, I was interested in pursuing a PhD in cancer biology. And although I did have a few PhD offers, I had to turn them all down because none of them could offer me full funding. As an international student, it was really difficult to have to be able to fund even part of the PhD because of increased international fees and high conversion costs. So Simon was aware of my aspirations, and he took it upon himself to help me with my PhD. And I'm very grateful to him because he helped me secure part funding for my PhD through the University of Nottingham as an international special scholarship. However, this wasn't enough to fully cover my international fees and stipend and etc. And it is then that LAM Action very gratefully came forward to fund all my remaining expenses and finally enable me to fulfill my dream. So I would like to take this opportunity to say a big thank you to LAM Action for raising the money for research that has helped fund my PhD, and also to all donors, LAM donors that have been able to kindly donate their tissue, which has been such a valuable resource for my PhD, and without which I would never have been able to do most of my research that I have done. So now, funding secured, I then began my PhD titled Modeling LAM in 2D and 3D co-culture systems. So we already know that cysts are formed near LAM nodules in the lung, and these cysts are formed due to the destruction of the LAM lung, of the lung. Proteins known as proteases are capable of destroying organ tissue, and these proteases could possibly destroy the lung-forming cysts. Now, a LAM nodule consists of varying type of cells, a few of which carry the TSC2 mutation, but a large proportion of them are spindle-shaped cells that do not carry the mutation, and these resemble cells known as fibroblasts. And fibroblasts are cells that are already known to produce several such proteins known as proteases. So knowing this, my PhD aims were to identify which proteases were increased in LAM, to design a lab-based model to determine the cellular interactions, effect of cellular interactions on such proteases, and then to investigate the effects of estrogen and rapamycin on our findings. So initially, I wanted to find out what are the proteases that would be involved in LAM. Just a few months into my PhD, I was able to receive tissue from lung transplant, lung transplant donated by, kindly donated by one of our LAM donors. And with these tissues as a resource, I screened for several proteases to look at which one was highly increased in the LAM tissue. And I found that a protease known as cathepsin K is highly increased in LAM as compared to normal lung. So I then wanted to find out and confirm that cathepsin K is present in the LAM lung. I found that cathepsin K is present in LAM nodules, seen here as brown coloring, and in the normal lung, you can barely see any presence of this protease. So as we already know that spindle-shaped cells of fibroblasts are present in the LAM nodules, and we have been the first to report that, and it is also known that fibroblasts are cells that can produce these proteases. I wanted to find out whether the spindle-shaped cells seen in the LAM nodules or the fibroblasts from the LAM nodules also produce cathepsin K. So, and I found out that they do. This is an image of fluorescent staining. FSP, or fibroblast surface protein, is a protein that identifies fibroblasts, which is green over here, and cathepsin K has a red stain. And when these two proteins are expressed by the same cells, or they're produced by the same cells, the merged image shows a yellow coloring as seen here. And these cells are elongated spindle-shaped cells which were identified to be fibroblasts, also producing the protease cathepsin K. However, the mere presence of cathepsin K is not enough to destroy lung tissue, and cathepsin K is a protease that needs to be activated, and it can only be activated in an acidic environment, that is at low pH. And therefore, I grew my cells at different pH scales. pH is the measurement of acidity, and a pH of 7 and below gives a more acidic environment. And when I did that, the LAM-associated fibroblasts, as we term them today, which are the fibroblasts that are from a LAM lung, had an increased cathepsin K activity at pH 6 and 7. However, this activity is further increased when the LAM-associated fibroblasts are grown together with LAM cells, which are the cells that carry the TSC2 mutation. This suggested that these two cells support each other and possibly have an existing symbiotic relationship. So then I wanted to find out whether it is the LAM cells that provide a perfect environment to the LAFs or the LAM-associated fibroblasts to produce and activate cathepsin K. I then grew the LAFs on their own, and over time they do not show any change in pH. However, when I grew the LAM cells, they dramatically reduced the pH over a short period of time, and the addition of rapamycin inhibits or halts this pH change. So then suggesting that it is the LAM cells that provide an acidic environment for the LAFs. I then wanted to, so now we knew that the LAM cells and the LAFs support each other and to help with disease progression. We therefore wanted to look at this mechanism in a more systematic manner. So to do so, we analyzed the effect of LAM cells on LAM-associated fibroblasts and normal human lung fibroblasts in order to find out if there are any similarities or differences between the two. And we did this in such an experimental setup with the fibroblasts at the bottom and the LAM cells in the top, and the effect of this was measured in the fibroblasts. To do so, we analyzed all of the genes from the LAM-associated fibroblasts and normal human lung fibroblasts using what is called as a gene chip. And we got several results out of this analysis, and one of which you can see here. This is known as a heat map, where the varying colors, the different colors, show the varying quantities of the genes that are affected or that change because of the presence of LAM cells. And these genes are from the fibroblasts. So what we found out from these analyses were a list of proteins that are signaling proteins known as chemokines that were highly elevated or increased in the LAM-associated fibroblasts, about between 2 to 40 time increase when these fibroblasts are in the vicinity or when they are near LAM cells. And these chemokines were also then found to be present abundantly in the LAM nodules from our donors. Now, chemokines are signaling molecules that signal to various immune cells. And therefore, we looked at whether immune cells are present in the LAM nodules as well. And we did find the presence of neutrophils and mast cells. These are two types of immune cells that were found in our LAM nodules. This is important because immune cells are also other cells known to produce several other proteases, which could then result in further degradation of the lung tissue. So, to summarize all of my findings, we have already shown the presence of fibroblasts in LAM nodules, but what I found during the course of my PhD was when the LAM cells and the fibroblasts are near each other, the fibroblasts result in the production of cathepsin K. This cathepsin K can then be activated due to the acidic environment provided by the LAM cells. The activity results in the degradation of tissue, of the LAM lung tissue. And the fibroblasts also in the vicinity of the LAM cells produce these chemokine signaling molecules that then attract various immune cells, and the immune cells then infiltrate into the LAM nodules, thereby leading to further destruction of the lung. So I have now come to the end of my PhD, but there are a few unanswered questions which, when answered, I think would lead to very exciting findings within the LAM research. So I would like to identify what causes the LAM cells to acidify this environment. We already know that proteins known as membrane transporters are responsible for acidification, and whether blocking these membrane transporters with inhibitors that are already available may decrease lung destruction. There's an inhibitor of cathepsin K, which is already a trial drug. So the addition of this drug, the use of this drug, whether it would be beneficial again to treating LAM, and also to identify the exact role of immune cells in LAM and how they may contribute to destruction, and whether blocking their recruitment using a chemokine blocker would be again useful, which again is available as well. And I would then firstly like to thank both my supervisors, Professor Simon Johnson and Dr. Debbie Clements, for their excellent guidance. I would like to thank all the LAM patients who have donated their tissue, which has been an extremely valuable resource for my PhD. And I would like to thank LAM Foundation also for to help with some of my chemokine research. But lastly, I would like to thank LAM Action, who has very kindly funded my entire PhD and has made it possible. And I would love to continue in this field if possible, because I think our findings one day will be very big in the LAM research world. Thank you.
Hi, everyone. I'm just going to carry on from Aru's talk. And what I'm going to do is really give an overview of one particular aspect of what we're working on, and that is lung destruction in LAM. And really what I want to talk about is why we're going to take the direction we're taking at the moment, what new ideas we've had, and why that's really modified our research a little bit over the last couple of years. But before I start with the new ideas, I thought I should kind of highlight what we thought was happening in LAM, and then I'll tell you how it's changed and what the implications of that are. we used to think of LAM in the lung as a little collection of cells which was destructive, and we knew they had a mutation in a gene called TSE2. Because of that, they didn't make a protein called tuberin, and we thought all the cells in the lung that were causing the destruction were these LAM cells. And this was fine, because actually it was a very productive time. We know what tuberin does. It's involved in sensing the environment of the cell, and then passing a signal on to other proteins, which then tell the cell whether to divide. If conditions are good, there's plenty of food around, or actually just to hold off for a while if conditions are a little bit rough at the moment. And if you don't have tuberin, what happens is this process gets all uncontrolled, and the cells grow when they shouldn't grow. Because tuberin was identified as a component of LAM, that led us to identify mTOR as a target, and there's, as you know, an inhibitor of mTOR1, which is rapamycin, and is an effective treatment for LAM. So that's all great, but what was happening with us was we were finding that our model of what LAM was changing. We started to realize that it wasn't just cells with a mutation that were present in the lung. There were other cells there as well. Our favorite cell that's also in here is the fibroblast, but there are other cell types there as well that we're also working on. And this led us to ask some other questions. Where are these cells coming from? What type of cells are they? We have to identify what they are. But more importantly, are these cells responsible for this destruction you see in LAM? So is it not just the cells that have the mutation that we've been working on for so long? Is it these other cells that might also be involved? And that's really led us to two main new ideas in LAM, and this is the first one that I'm going to outline for you today. And that is, it's not all the LAM cells' fault. Some of it is because of the cells that they're bringing in. So this is a LAM cell. These are cells that are around everywhere in the body. They're called fibroblasts. They're generally well behaved, and they're very useful, and they produce small amounts of what we call matrix degrading proteases, which helps them to do their job, but that's all fine. When LAM cells arrive in the lung, what happens is they send a signal. And what this signal does is it causes the fibroblast to huddle around the LAM cell. We think that this has a benefit for the LAM cell, which I've talked about before. But what the LAM cell also does is it causes a change in the environment around itself. And unfortunately, this change causes these proteases produced by the fibroblasts to become hyperactive. They're much more active than they should be. So now we have a double whammy. What we have is a whole load of these fibroblasts in one place, and the enzymes they're producing are very, very active. So you can see now we have a problem, and this could potentially be a very destructive situation. So what can we do about this? Well, what we do is we identify all the pathways involved, and then we use existing tools to stop them from working. First of all, the protease. If there's an inhibitor, we can stop the protease from working immediately. That's fine. Can we stop the LAM cell from making this little environment around itself so that the protease won't be so active? So yes, we can do that. And the thing that we're really aiming to do is, can we stop the LAM cell from recruiting the fibroblasts in the first place by understanding more about what this recruitment mechanism is? So that's what we're doing. So then we get back to the normal situation. The LAM cell can't recruit the fibroblasts, the fibroblasts can't produce the protease, and everything, and the lung destruction stops. And the best example of this is what Aru's just highlighted from her work, and that's cathepsin K. We know that fibroblasts produce cathepsin K. It's what they do. It's part of the enzymes that they make, and that would be all fine, except LAM cells attract fibroblasts. So then they all form this little clump in the lung, and there are loads of fibroblasts all in one place that really shouldn't be. The other thing to do is LAM cells make their environment acidic, and that's a common thing seen in tumour cells. And unfortunately, cathepsin K is much more powerful under acidic conditions. So you can see we have this double effect. We have too many cells, and we have an environment that makes these proteases work extra well. So the new idea we have, the new idea from this, is that it's not one cell type that's causing the problem. It's the combination of the two cell types together, each of which contributes to part of the problem. And cathepsin K is the best example we have at the moment, but there are other proteases that we're looking at and other cell types that will probably have the same thing. So this is one aspect of the research that we're doing that Aru's highlighted, and actually there's a poster outside that details this work. The second new idea that we have in lung damage in LAM is that LAM cells, just by being in the lung, make the area around them more fragile. And this is one way we think that might work. Here's the LAM cell, and here's a fibroblast. And we know from the experiment that Aru's done, where she's analysed all the genes that the cells produce when they're together, that fibroblasts receive a signal from LAM cells and they change. It changes the genes they express. They make things now that they didn't make before, or they make some higher levels of some things that they already made. And one of those things that we found they make a lot more of when they're with LAM cells is a protein called FGF7. FGF7 is quite an exciting protein for various reasons. I'll explain in a second, but what we find is that LAM cells themselves hardly make any FGF7 at all. And fibroblasts on their own make some, but when you put the two together, you get huge amounts more FGF7. We can measure how much more by just measuring the protein that they put into the medium they're growing in. And we know, actually, that if we treat the LAM cells with rapamycin, they get less good at making the fibroblasts make FGF7. So we can kind of stop them from inducing FGF7 in the fibroblasts, which is good news. So why is FGF7 important to us, and why do we think it's so interesting? The first thing is it makes your hair grow, apparently. Every time I look for FGF7 on the internet, there's some random Japanese company that tries to sell me shampoo. I don't know if it works, actually. It might do. There is good biological evidence that it might, but there you go. We're more interested in what it does in the lung. So this is the important bit of your lung. This is where gas exchange takes place. This is the alveolus. So the air comes in here and it exchanges with the blood in the capillaries. And there are two types of cells that make up the alveolus. There's these super thin ones here, which you may not be able to see, but all this is made up of really thin cells. And there are these little fat ones. The thin cells are kind of the structural ones, and they're called type 1 cells. And the little fat ones here are we call type 2 cells. And luckily we can find these type 2 cells in the lung because they helpfully make a protein called SPC. So if we look for where the protein is, we find the little fat cells, the type 2 cells scattered here and there. There's one helpfully arrowed. What we do know is, and we've known for a long time, is that the little fat cells, the type 2 cells, absolutely love LAM nodules. So here's a little nodule of LAM cells in the lung, and it gets absolutely plastered all over with these type 2 cells. We've never really known why. It's just been something we find in our histology, and we've never really thought about it. Sometimes, I don't know if you can see here arrowed, the type 2 cells actually get within the nodule itself. They love the LAM nodules so much. We think, so this is a model of what we end up with. We end up with the LAM cell, and around it the fibroblasts, and around it the type 2 cells. And the reason these are here is they love FGF7. So the LAM cells are causing the fibroblasts to make FGF7. The FGF7 spreads out and causes these type 2 cells to come to the LAM nodule. So this is my little cartoon of what it looks like, and this is what it looks like if we look at it by histology. Here are the little cells plastered round. So, you know, why would this be a problem at all? So LAM cells cause fibroblasts to make FGF7. FGF7 attracts the type 2 cells. The type 2 cells are very important in the lung. When the type 1 cells wear out, the type 2 cells replace them. So the type 2 cells actually make the type 1 cells. And they do this all the time. This is their function. They are what's called stem cells for the type 1 cells. That's what they do. But now we have a problem, because now all the type 2 cells around the LAM nodule are wandering off and splatting themselves to the outside of the nodule. What we think is happening is because there's a LAM nodule, all the type 2 cells from around it are coming and they're migrating and they're coming into the nodule and they're going away from where they're supposed to be, where they're supposed to be doing this repair function of replacing the type 1 cells. And what that's going to do is the whole area around the nodule now just might be a little bit more fragile. It might be a little bit more delicate because it can't be repaired as effectively because the cells have wandered off because they love FGF7 that the fibroblasts are making. So these are our 2 new ideas in LAM. The first new idea, which is probably 2 or 3 years old for us, is that there are different types of cells in a LAM lung, not just the LAM cells. The cells talk to each other. We know that they communicate. We've identified some of the signals they send to each other. We know how they transmit the signals, and we know how to block a lot of the signals. Because now you have cells together that really aren't usually together, you start to see some unusual effects. So you start to see proteases that are more active than they should be, or cells wandering around and ending up where they shouldn't be. We think that these things contribute to the lung damage in LAM. And we do have tools that can stop this happening in the lab, in our experiments. So the important thing is these are actually quite new ideas for us, new ideas that we've applied to LAM. But they're not entirely new in, for example, cancer research or cell migration research. And because of this, because these are pathways that have been studied a lot by other people in other contexts, in other diseases, that's given us a lot of new tools, a lot of drugs we can use, a lot of drugs we can test in our models to see if they would be effective in LAM. So this is the direction that we're going to take our research in, at least part of the direction that we're going to take our research in. And it's early days, but Aru's made huge progress in her PhD in identifying the signals and the receptors and looking at an inhibitor. So we're really optimistic about how this is going to work out. So my last slide is obviously just the acknowledgement slide to the LAM patients that have donated tissue to our study, to LAM Action for funding our research for so long, and also to the LAM Foundation, who specifically funded a transcriptomics study last year, which has really underpinned a huge amount of the research that we're doing at the moment. So I'd like to say thank you, and if anyone has any questions, I'm very happy to answer them.